So next up is another of our international invited speakers. So it's my real pleasure to introduce Martin Kircher, who's travelled over from University of Lübeck in Germany. Thank you so much. And Martin's going to be talking to us about interpreting variant function across the human genome.
Yeah, thank you so much for inviting me and making me part of this.
Can everyone hear at the back? Is that a yes? It's the cryptic color code. I think it needs to go green. Yeah, it should be green.
Green it is. I don't know. Can we? Is that better? A little higher now? Yeah. Can try. So thank you so much for inviting me and making me part of this inaugural symposium here. I think functional genomics is obviously a very important topic. To start out with, and I think we had the motivation before, right? We're sequencing more and more genomes to identify disease causal variants. But what does that actually mean? Well, if you do an exome, right, it's still a somewhat manageable number of variants. But if you do a genome, you're talking about 2 to 4 million variants per individual. And then if you apply just an allele frequency filter, you're still probably stuck with something like 150,000 variants that you would potentially need to interpret. And so if you're a clinician, then you would be walking through those variants in a hierarchy, right? So you would look first for, let's say, nonsense variants, and you would be looking for amino acid changes, canonical splice site changes, and so on. And you would be walking through these classes, and you would be using within each class additional annotations that you might find and try to prioritize variants within each class. But you end up sifting through a very large group of variants. So what you would like to do in an ideal world, you either have computational methods that assist you, or you have high-throughput functional genomics assays that allow you to scan these sets, and for the later ones, preferentially in a preemptive way. So you have done that at some point. You scanned all potential variants, and we've heard some really cool work from Leah about that for coding sequences. So in my presentation, I would like to touch on both things, so the computational as well as these experimental approaches. And for the computational, this is actually the longest running project that I've been involved in. So it's been running since my postdoc times with Jay Shanduri. We've developed CAD, then also when I became an independent PI in Germany, and I had my first PhD student on the project, and then another one came and expanded it to structural variants. And then a number of other people from the lab have contributed since. So about that. So getting back to these variant hierarchies and how to use them for prioritization. If you now take these annotations and you look at them across the genome, you quickly realize that some of these annotations or some parts of the genome have really a lot of annotations. Others are very sparse. You have very different kinds of annotations. You have categorical annotations, so it falls within a block or it doesn't fall within a block. You have quantitative measures, how well is something conserved across species. And it is really hard to handle, so to walk through. So if we had a labeled data set of disease causal variants and no effect variants, right? We could train a supervised machine learning model, and we could present it with all these annotations, and we could leave it to this model to learn how each of these annotations relate to the labels, and we would be more flexible, we would have less of a bias, and we would even get a measure of confidence for each of those variants that would be calculated out of these annotations. So how does one actually get such a labeled data set? When we started, there was no ClinVar. And it would also have been too small and too biased for us with its focus on coding effects. So 10 years later, we thought, well, maybe it's a worthwhile question to ask again, would ClinVar be useful for us for such a challenge? Because as Leah has already shown, ClinVar has substantially grown from its 27,000 variants that it had in 2013 in its initial release. So if you look at the whole genome as it's, or at all potential variants of the genome, as seen here by essentially ensemble variant effect predictor, that's the distribution of categories that you expect. This is ClinVar, and so what you can see is that there's a large overrepresentation of these severe protein coding effects. If you actually split this even further and split that into pathogenic and benign assertions, you see that essentially those regulatory variants, they're only present in the benign. You see barely any pathogenic ones. And this class of intergenic variants is completely missing here. And now you might wonder, well, maybe this has changed in more recent submissions to ClinVar. And the unfortunate answer is no, right? This proportion has stayed the same. So you have this very clear underrepresentation of non-coding variation that also didn't improve over the years. So what we came up with 13 years ago is actually still very sensible. So we used variants that changed on the human lineage since the last common ancestor with chimpanzee as a benign neutral set because it has seen so many generations of purifying selection. And we contrasted this with essentially simulated de novo variants. So we just drew variants across the genome with the same substitution pattern. And we contrasted those. And so if you contrast these proxy benign with the proxy deleterious, you can actually see how purifying selection created this annotation-dependent depletion. So here you see it for categories produced by ensemble variant effect predictor. And you see that there's a 20-fold underrepresentation of stop-gain variants, for example, in our human-derived versus our randomly drawn mutations. So that meaning, right, that purifying selection just causes variants to be missing from constrained regions. So if you want to get from a single annotation dependent to this combined annotation dependent depletion, which we then called CAD, is we take these annotations, we strip off the coordinates, we strip off the gene identifiers, and we put all that into a supervised machine learning approach. Actually, a rather simple model, a linear logistic regression here, which we extended by interaction terms for base substitutions and amino acid substitutions, as well as some interactions between the predicted consequence and some of these, let's say, conservation metrics. So you take essentially these 80 diverse annotations and you plug them in, you calculate one score. So this is in a nutshell what CAD is about and what has been actually widely adopted by the community over the last decade. So I want to do a brief mention because I said, well, you can't use ClinVar, but actually something has changed these days. You could be using Nomad. We have a preprint if you're interested in that. So you can actually train similar models using singletons versus common variants. You even get a data set that is slightly larger than the one that CAD uses for its training. Another side note that I wanted to make is that also now, 3 years back, we've extended this basic concept of CAD, and I have to say structural variants are a very different beast, right? The calling of their positions is very imprecise compared to the short variants that CAD is dealing with, and you have these larger genomic segments that you need to aggregate annotations across. But we've developed a tool on the basic ideas of CAD, also for structural variants. And yeah, feel free to use it or to read the manuscript. Okay, so back to the results and how CAD actually gets us away from these hierarchies of variant effects. So if you, for example, look at the gain of a stop codon, so these nonsense variants, they will depend on where they happen along a gene and in which gene they happen. So consequently, we get a broad range of scores of potential nonsense variants, as you can see here on the bottom of this page. And so you can generalize this and you get a combined metric that integrates various annotations and gives you scores for single nucleotide variants, multinucleotide substitutions, as well as small insertion deletion changes. So we get to see variant effects on a continuous scale and integrated across molecular effects, for example, having a splice effect coming together with a missense effect. So you get this integrated into one measure. Okay. So at this point, you're probably wondering, why is he talking about something that's 13 years old? Because we try CAD to not age. So we try to update it every few years. And so our last update was actually just last year, where we released version 1.7. And so what did we try to do there? Well, we first of all always update the underlying transcript and gene models. But we also try to look for new annotations that might be useful for using CAD. And one of the big things that we identified with this last release was that we wanted to include sequence-based models and their predictions. And I want to talk about two of these models that we integrated there. So what does that mean to integrate sequence-derived scores? Well, there are models where you can plug in a sequence, so you put in your reference sequence allele, and you plug in your alternative sequence allele, and you get two scores out of these models. You take the difference of these scores, and you put the difference into CAD. Right? So that's what this means. And so the one thing that we integrated was this meta-ESM1V model. And so this is a large language model of protein amino acid sequences. So it takes about 1,000 amino acids as a text, essentially, and it has learned from, from hundreds of millions of naturally occurring protein sequences, which amino acid is usually neighbored by which, how certain folds might look like, right? This is what it intuitively then learned to represent in its structure. So you can take your sequence, and in the middle, right, you can then ask how likely is another amino acid at this position. That's what the model gives you. And so others had benchmarked this and had seen very good performance on deep mutational scanning data, as you've seen from Leah earlier today. And so we said, okay, let's integrate it. We integrated it, and we also went through quite some additional effort to also make it work for nonsense and frame shift variants, which the model by default doesn't support. But as you can see, the gain is small. But measurable. So we thought, well, maybe not too surprising. Already quite a few years back, we had noticed that actually coding predictions, they're approaching some kind of saturation. There's, the models are already pretty good. And so we thought, well, let's move on. Let's try regulatory sequences. Okay. So regulatory sequences. what people have been doing there for more than 10 years now is they have tried to use advances in object recognition from images, trying to apply those to DNA sequences, and essentially have these rather sorry, I'm doing something wrong when I press the pointer. I don't press the pointer anymore. Okay, you have these very narrow images. Maybe I can actually do this. Right, you have these very narrow images that are just long, like the DNA sequence. Yeah. And so a diversity of tools have been developed over the years, and they've become more and more complex. And what they all do is from sequence, they're trying to predict different functional readouts of sequence. So this could be ChIP-seq peaks, DNase, attack peaks, or most recently also gene expression. And they've been doing so with a wider and wider sequence context and with more and more potential outputs. So when we started on this endeavor, we thought, well, Informer was the state-of-the-art model at this time. Let's try to use that, and we realized that the computational burden was just too high. We could not run that for the whole genome and all potential 9 billion substitutions that you can do to the human genome. So we went for a much smaller architecture, a convolutional neural network, just predicting 6 different cell lines as output, just the open chromatin state of those, and we integrated that. Now again, I have to say, you see some nice effects where our model started to also recover a region of where we had previously done MPRAs and determined activity, but overall the model performance barely changed. Right? So, and it took us more than 2 weeks on GPU resources just to calculate this whole thing. So, just so you know. So I hope this was not too negative in terms of investing resources for what seems to be a little performance gain, because I think the actual message here is that we are gaining in performance. So taking in additional annotations improves CAD, and we will continue to maintain it, and we will continue to look for annotations. Some notes about these specific models, right? They typically measure a certain molecular function, and it's important to understand that they only measure this certain molecular function. And they also do not work for all variant classes, which leaves then substantial work for their integration in addition to their high computational costs. For the future, we really have to think how we can capture cell type specific effects in CAD, which right now CAD is an organismal model, so it's not supposed to do that. And we probably also need to think about how we better capture nonlinearity when integrating old and new annotations. So this is where I want to switch a bit into the experimental side and how we measure and model especially non-coding effects. And now, non-coding is such a horrible term because it just means, well, the opposite of coding. And the opposite of coding is a lot of different molecular effects. And so I want to be a little more precise. Whenever I talk now about non-coding, I talk about transcription initiation, I talk about promoters and enhancers, but there's a whole list of other things that are non-coding. It's also not just since yesterday that we know that regulatory sequences are important. We now know for more than 50 years. The one thing that has changed is that we now really have these large catalogs of potential regulatory elements, and we know where they potentially reside in the genome. And as Wendy has also beautifully explained this morning, we have a much better understanding how promoters and enhancers interact by chromatin looping. So over the past 15 years, various of these multiplex assays of variant effects, as also Leah was talking about, have been developed, and especially also some for regulatory sequence effects. And with our collaborators, Nadav Ayitouf and Jay Shenduri, we are especially using so-called massively parallel reporter assays or short MPRAs. And if you've seen a luciferase assay, which I assume many of you have, just imagine it in a multiplexed readout of RNA and DNA abundance rather than reading the light. Yeah. So in a nutshell, what you do is you first define the sequences that you want to test. Frequently, you do that by oligosynthesis. You add unique barcodes or tags to those sequences that will later allow you to quantify that specific sequence. The sequences are integrated into a reporter construct. This reporter construct typically has a GFP, even if you don't want to read out the light. And these plasmids are then either directly brought into cells as episomal plasmids or genomically integrated, for example, using lenti. After a few days, you then collect RNA and DNA, and you amplify out these barcodes or tags. You count them, and you measure activity as RNA over DNA abundance of the barcodes. I also put some pros and cons here, and to quickly summarize, these are really nice high throughput experiments, but you have to realize that you're doing it in a very artificial system. Yeah. So you're not testing the sequence in its native context. As you probably appreciated by now, my group is computational. So we've been developing some key resources for the analysis of such data sets, but we've also been contributing to the modeling of such data. And I want to highlight a few of those things. So one example here is from our colleagues in San Francisco, where they tested candidate regulatory sequences that were identified based on open chromatin marks and then tested for 3 different cell types. As you can see from these numbers here, it is possible with these assays to test 100,000 elements in a single experiment. You can look at different things, of course, and learn different things. And something that we learned specifically from this data set was very interesting for me. And it was just that these 200 nucleotides of core promoters that we put in with otherwise enhancer regions, that their MPRA activity very well predicted the steady state expression in the cell type. So this highlights again, I guess, importance of promoters as main regulators of transcript abundance. Another thing that we tried to do here was we tried to predict the measured MPRA activity from other information. So could we have predicted our experimental readout? And we tried that by using peaks. So these are well-characterized cell lines. So for example, especially, for example, HepG2 or K562. And so you can ask, how well do these chip measures of the genomic locus that we have predict the activity in the MPRA? How well does a convolutional net, like the one that I just presented for CAD, predicts it? And how well can it be predicted from the outputs of Informer? So if you take the outputs of Informer and build a new model, what predicts it best? And one might have suspected it, but the most complex, the Informer model, predicts the activity best. Actually, to extent that it achieves or approaches the experimental replicate correlations. But the simpler DNA sequence model is already pretty close with much less computational costs, and it actually allows us to go into the model and then look for TF binding sites that are driving these activity signals. And we can then also go for signals between the cell types and look for cell type specific effects. Okay. Instead of those more classical MPRAs, we can also do other studies with MPRAs. And here, for example, we tried to study how promoters and enhancers act together. And we've already heard about capture high-C approaches as mentioned in some other presentations. But so this is proximity ligation, and then a targeted enrichment for a little more than 500 promoters. And so what you get is you get long fragments in this case, so approximately 1 kb, and part of that sequence is a promoter for which you've been enriching. And then you put this into an MPRA, so you get both information. You get the high-C contact information from these fragments, from sequencing these high-C fragments, and you get an MPRA information from plugging these fragments into the MPRA outline that I just gave you before. So the proximity ligation has this feature that it also creates you a bunch of fragments where you only have the promoter without any enhancer, and you can use that as your background measure of how much variability you have in this promoter sequence depending on its fragmentation. And then you can see at which point an enhancer sequence actually significantly enhances or represses this promoter, right? And we can call upregulating and downregulating elements. And we know that weak promoters seem to have more enhancers, while strong promoters seem to have more silencers. And what is even a little cooler, I think, is that we can also look at cases where we have the same enhancer interacting with multiple promoters. And we can now ask whether we always get the same effect, right? Does this enhancer that activates promoter 1, does it also activate promoter 2? And as you can see from these heat maps behind me, that's not the case. You can also have a promoter that activates one gene and represses another gene. And we looked into this a little more because you might be wondering, why does it happen? And we think it's actually certain pairs of TFs that come together between the promoter and the enhancer. And some of these pairs are then repressing and other pairs are enhancing. And so these are the significant pairs that we identified in this analysis. And yeah, this is actually something which we just released as a preprint on Saturday. So please feel free to have a look if you're interested in that. Okay. And then I guess I'm running out of time, so I spare you the details of the rest. I would have 2 more studies that we did within the IGVF project, and I'm going to directly go to conclusions. So I guess for the first part of my conclusions, this is really what also a lot of other speakers have shown today. Understanding variant effects is complex and always involves thinking about multiple molecular mechanisms that might be coming together. Secondly, these experimental perturbations at this point are really critical for us to understand molecular processes that we don't understand well, like how does non-coding sequence work, right? How does regulatory sequence specifically work? And then maybe some take-homes like element activity for regulatory sequences works well, variant effects do not. And MPRAs, I guess, have a good power of detecting activating variants even on low activity elements, while if you want to look for repressing variants, you first of all need some activity to actually then also measure its reduction. So big thank you to our collaborators, without whom we would not be able to do this kind of research that we do. And obviously the biggest thanks to Jay and Nadav's lab here. And then thank you to my lab.